Hi guys, me again. This video will be for April the 13th, a Wednesday, for my Conceptual Science classes. So hi to all you guys. It's nice to see you, even if it is only just this way. And today you should have Miss Pettit. I hope I'm pronouncing your name correctly, but hi there, how are you? Thank you for helping us out today. We really appreciate it. So today we're going to go through slides. We're going to move on to talking about DNA, and that's going to take us till the end of the year. This is the last section that we will be doing. And the handouts that you'll need for this, they're in the office area that's attached to room 200, and it should be labeled appropriately with April 13th for conceptual. I think they were actually copied in two pieces, so there might be two handouts you have to give out today. And then there's another stack for a homework assignment that you can distribute today as well. And also, I'm going to record this video and go through all the slides, but I highly doubt that there'll be time to get through all the slides. So however far you get is fine. And when we come back, or when I come back after my chemo week on the, I think it's the 19th that I'll be back, we'll finish up however far you got. We'll finish up the slides on that day. So in the last video I did, you guys know I was still coughing a lot. Well, my wife brought me a shaved ice from Bahama Bucks. Just as I say that, I was about to cough. So I'm going to try to be snacking on that because that feels really good on my throat. And maybe that'll help keep me from coughing so much in this video like I did in the last one. So I think that's all I have for intro. So let's move on to the slides. And just like I said in the other one, I'm just going to go through these at a normal pace, and you can stop and pause it however much you need to make sure. Dang it. There we go. Well, hopefully we won't have as many as I had last time. Anyway, and you can stop and pause it as much as you need to let everybody get caught up with the fill in the blanks. So let's switch to the slide view now. There we go. So DNA is absolutely amazing. It is unlike anything else, any other molecule. It is unbelievably unique, complex, organized, choreographed, controlled, structured, arranged, all those things. Yet at the same time, it's so simple. It's only composed of 4 building blocks. It stands for deoxyribonucleic acid. It's composed of carbons, hydrogens, oxygens, nitrogens, and phosphorus atoms. So it uses all the atoms that are commonly found in life, but its structure is absolutely unique. It is unlike anything else, and its structure is absolutely required for life. And its functions are unique, and it governs and performs all the processes required for life. On top of all that, it's a molecule that stores information. But not just that, it's a massive amount of information. This is another area in which DNA is unique. This information is incredibly complex, organized, and logical. Yet, like I said, it's so simple because it's made up of only 4 building blocks. But it not only stores this information, it utilizes it to create, develop, grow, sustain, reproduce, protect, and heal life itself. Life depends on DNA structure, its functions, and its storage and utilization of information. So you could say DNA is large and in charge. And I just did say that. So if you don't have DNA, you don't have life. It was discovered in 1953, and the credit went to James Watson and Francis Crick, although there's other stories about the contributions of people like Rosalind Franklin who didn't get the credit, but I'll leave that for another discussion. And it was heralded as the discovery of the language of life, which is correct because that's exactly what it is. I told you it's made up of 4 building blocks. Those building blocks are called nucleotides. And each nucleotide is composed of 3 groups. A nitrogenous base, which just means it contains nitrogen, a sugar called deoxyribose, and a phosphate group. A phosphate group is PO4, so phosphorus and 4 oxygens. All the chemical bonds in each nucleotide are covalent bonds. And if you remember, that's a bond where there's equal sharing between the atoms. So these are covalent bonds that hold it together. If you drew a 2D picture of a nucleotide, this is what you would get. They're showing the 3 groups, the nitrogenous base, the deoxyribose sugar, and then the phosphate group. And the name of that would be 2'-deoxyadenosine monophosphate. We'll get to that in a minute. So that's what a nucleotide looks like. And there are 4 different types of those. There's adenine, which everybody just abbreviates these to the single letter, which is A. Guanine, which is G. Cytosine, which is C. And thymine, which is T. Those 4 letters, A, C, G, and T, make up the 4 letters in the alphabet of the language of life. The difference between the 4 nucleotide is the nitrogenous base group. So here is what A, G, C, and T look like. And we go back to this. My pen's not working again. There we go. All you do is switch that out. So this one happens to be A. But if you switch that out for the other 3, that's how we get G, C, and T. So if you replace it with any of these other 3 in that same location, we end up with G, C, and T. And if you notice, their structures are not all the same. But you also see that A and G have 2 ring structures, and T and C only have 1. So based on those structures, A and G are called purines, and C and T are called pyrimidines. So the ones on top are purines, and the ones on bottom there are called pyrimidines. This is what I just said. Guess I got ahead of myself. By changing that base, you change it from an A to a G, C, or T. And we end up with our 4 building blocks of DNA. The positions of the atoms in the ring structures of nucleotides are numbered. For the deoxyribose ring, a prime is added to the numbers to distinguish them from the numbers in the atoms in the ring structure of the nitrogenous base. So what that means, down here, 1, 2, 3, 4, and 5 prime, all of those, these will all be carbons, by the way. All of those positions are given 1, 2, 3, 4, and 5 prime. The numbers that go in this group, oh, no, I just dropped my pen. The numbers that go in that group do not get primes. Let me back up. This is what I was showing here. So in this position of the nitrogenous base, notice these numbers here do not have primes after them. That's how we distinguish whether we're talking about the positions in the base, the nitrogenous base up here, are the positions in the deoxyribose sugar. These have primes, these do not. And if you look down at that deoxyribose sugar, that's where DNA gets its name. In RNA, which we'll talk about next, it uses a typical sugar, a typical ribose, and in that typical ribose, at that 2' position, it has an OH. So for RNA, this guy here would have a OH. But for DNA, notice it's missing the O. It only has an H. So it's without the O, or it's deoxy. It's without oxygen ribose. So that's why it's called deoxyribonucleic acid. And that's why RNA is called ribonucleic acid. I throw that in for free. I won't charge you any extra for it. And if you compare them side by side, here you can see the difference. So the one on the left would be used in RNA, and the one on the right would be used in DNA. And again, this is what I've been showing you a couple slides already. The ring atom positions are numbered with the nitrogenous bases too, but there's no prime after the number to differentiate between the sugars and the bases position in the nucleotide. If you have a sugar molecule attached to a base without a phosphate attached to it, it's called a nucleoside. And each nucleoside has a unique name. So A is adenosine, G is guanosine, C is cytidine, and T is thymidine. So if you see that nomenclature, it's talking about just the nitrogenous base with the sugar. So nucleotides are named according to their nucleoside, the base and the sugar. Which ribose sugar is used, and which ribose sugar is easy for you to say. Which ribose sugar is used, and then how many phosphate groups are attached. So with deoxyribose, deoxy is used in the name, and a small d is used in abbreviation to indicate that it's deoxy. But see, the thing is, there can be 1, 2, or 3 phosphate groups attached to a free nucleotide. If there's 1, it's called mono, 2 is di, 3 is tri. Same kind of nomenclature we had when we were talking back when we were doing the chemistry section. So now back to naming these. This is an A attached to a sugar, right? That's where the adenosine comes from. This is deoxy at the 2' position. This only has 1 phosphate, so it's monophosphate. So the name is 2' deoxy adenosine monophosphate. And if you abbreviate it, small d, AMP, deoxy adenosine monophosphate. But what if you have 3 attached? 3 phosphate groups, that is, right? See, there's 3 of them there. Everything else is the same, but now we got 3. So now this becomes triphosphate. So it's 2'-deoxyadenosine triphosphate, deoxy-ATP. The cellular energy molecule, ATP, is not this molecule. That would have the OH here. So this is a DNA nucleotide, and that's different from the cellular energy molecule that would have an OH there. So to make DNA, we take these nucleotides and covalently link them to one another over and over and over again to form a long strand of DNA. Just like Legos. And I'm sorry, I'm not going to do the demonstration today. But just like Legos can be attached one end to another, right? It's the same thing with DNA nucleotides. They just keep getting attached one to another over and over and over. And this can be millions to billions of times, depending on the size of the genome and the chromosome. The sugar and phosphate groups form the backbone of the DNA, and the bases stick out from the backbone perpendicular to that. But here's the thing, DNA is not just one long strand of nucleotides. It's actually 2 strands of nucleotides that are joined together in the middle. The uniqueness of this DNA structure and the essence of life itself is portrayed in how the 2 strands of DNA are held together. The two separate DNA strands are held together in the middle where the bases are. Each base sticks out into the middle and binds to the base across from it on the other strand. And there's a pattern to this. A and T always bind to one another, and G and C always bind to one another. And that binding of the 2 bases in the middle holds the 2 strands of DNA together. And that ultimately ends up forming a structure that looks like a ladder. The paired bases in the middle would be the rungs, and then the sugar and phosphates would form the sides of the ladder. But then this ladder structure gets twisted around like a spiral staircase or like a corkscrew. And therefore, DNA is called a double helix, because the 2 strands spinning around each other. And it's also called double-stranded. But the double helix lets you know that both strands are spun around each other. Now I told you the nucleotides are held together on each strand by covalent bonds. But in the middle, on the rungs where the bases bind to one another, A and T and G and C, that happens using hydrogen bonds, which are different than covalent bonds. It's the interaction of a hydrogen and an electronegative atom like a nitrogen or oxygen. If you remember back in chemistry, electronegativity is the measure of the tendency of an atom to attract a bonding pair of electrons. And the higher the electronegativity, the stronger it pulls. That's what all fits into how a hydrogen bond works. So DNA is basically 2 covalently bond molecules held together in the middle by hydrogen bonds. And if you were to, sorry, I was having some of my shaved ice. If you were to draw this out in a 2D structure, this is what it would look like. So here's the sides of our ladder, right? With the phosphorus, the phosphate, and sugar groups forming that. And then here's the rungs here in the middle, right? See that? Kind of makes a ladder. And this indicates where those hydrogen bonds are. And you notice with G and C, there are 3 hydrogen bonds. And you notice with A and T, there are 2 hydrogen bonds. Also notice that the G and A, the ones that have the 2 ring structures, bind to the C and T respectively. The ones that only have the 1 ring structure. So the backbone, look here, it's alternating phosphate, sugar. See that? So alternating sugar, phosphate, sugar, phosphate. The rungs are the bases sticking into the middle like I just showed you, with one base on one side bonded to the base across from it using hydrogen bonds. And in the order of the bases, if you read along this on one strand or the other, you would have a certain order of the bases. That's called the DNA sequence, where you just read the order of those bases like that and just list them as the letters. That's called the DNA sequence. Can't tell you how many times I did DNA sequencing in the lab. More times than I could count. Here's another image of that, how it's showing you how it's twisted around in the double helix. But then if you flatten it out, right? Same thing. We got phosphate, sugar, right? And then all of our bases in the middle, forming the rungs. So it's critical to point out that the bonds in the middle that hold the bases together, the hydrogen bonds, are not nearly as strong as the covalent bonds throughout the rest of the molecule. There's a big difference between those. Covalent bonds are much stronger than hydrogen bonds. So if we use some analogies of this ladder, so if it's a wooden ladder, actually, let me just jump down to this one. I like this one better. A metal ladder, where the vertical parts are held together with welds, but then where the rungs come together in the middle, each of the 2 halves are held together by magnets. The covalent bonds keep the 2 vertical backbone halves of the DNA ladder intact and are stronger and required for the integrity of the DNA molecule. So in this case with the metal ladder, we're talking about the welds. If you remove the welds, the whole thing's going to fall apart, right? It'll destroy the DNA molecule. Those are the covalent bonds. But the hydrogen bonds in the middle of the bases, they're weaker and they're not required for the integrity of the DNA. So like the magnets. So if you had 2 halves of a ladder held together by magnets, we could pull the halves apart, separate the magnets, and the 2 halves would still be intact. And we can put them back together and let the magnets come back together, right? And you could do that all day. And it doesn't destroy the molecule. All it does is separate the 2 halves. And that's exactly the way the DNA works. You can break those hydrogen bonds in the middle between the bases, and all it does is separate the DNA molecule into its 2 strands. These guys right here. We can split those open. We would still have this half, and we still have this half. They would still be intact. And we could put them back together again and let those hydrogen bonds reform, and the DNA would just be fine. Basically showing the same thing here. Oh no. Yes. Then I'll come back to this in a second. The other thing you need to know about DNA is that it has directionality. When you read the order of the DNA bases, like whatever it is, TGCC, whatever I have here, right? They're read and written in the order that they appear in the 5' to 3' direction of the backbone. And that resulting order of the DNA bases is referred to as the DNA sequence. Let me show you this. So here, on this side, notice that is our 3' position here. Here's our 5', 3', 5'. So what I'm talking about is you would read it in 5 to 3', meaning on this side we're going down. 5' to 3' would be going down, like this red arrow is pointing down. But on this side over here on the right, it's just the opposite. Because here's our 3', here's our other 5'. We're going 5' to 3', and on this side we're going up. So the directionality indicates 5' to 3'. If we read the sequence on this side, it would be going down. If we read the sequence on this side, it would be going up. So this is very short, but this would be CT, and this would be AG, because this one's going down, this one's going up. So notice they're aligned in opposite directions. On the one, which was the left one, if you go 5' to 3', you're going down. On the other one, which is the right one, 5' to 3', is going up. So each strand runs in the opposite direction of the other. I already told you A always binds to T and vice versa in 2 positions, so 2 hydrogen bonds. And G always hydrogen bonds to C, or what they call base pairs, in 3 positions, and vice versa. This shaved ice is really helping. Sorry if I'm making you hungry. It is pretty good. So where you find A on one half of the rung, or the one half of the DNA, you're going to find a T on the other one. And where you find a G on the one, you're going to find a C on the other one. And the 2 bases that bond together that create the rungs, they refer to those as base pairs. The abbreviation is a small b, small p. They call those base pairs. So these are base pairs, right? These are base pairs, etc. So when we have our magnets and our welds in place, we have our complete structure of DNA, what they would call as a double-stranded DNA. That would be what it would be called as its native structure. When we remove the hydrogen bonds from the middle, or the magnets on the ladder, you call that denaturing it. Remove those hydrogen bonds and separate the 2 strands. That's referred to as 2 single strands of DNA. So DNA is 2 covalently bonded molecules running in opposite directions of each other, held together in the middle by hydrogen bonds. So this is important because the nucleotides can only be added to a DNA strand in one direction. Just like Legos have a front and a back, right? They'll only go together one way, right? DNA has directionality like that too. The nucleotides will only go on in one way. And I don't know with the timing. We'll see if we get to discussing that process later or not. I just don't know. So when you get all your DNA put together, you start talking about the genome, the complete set of nuclear DNA that an organism has. And remember that eukaryotes also contain mitochondrial DNA, and plants have chloroplast DNA. So that's separate from the genomic DNA. So the genome does not include the mitochondrial DNA for eukaryotes and not the chloroplast DNA for the plants. We're talking about just that which is in the nucleus. And how much DNA there is depends on the organism. This guy right here, this marbled lungfish, this is the largest vertebrate genome known. He has a genome of 130 billion base pairs. Folks, that's a lot. 130 billion base pairs. Look at how cute this little guy is. A puffer fish. This guy, he has the smallest vertebrate genome known. He only has 385 million base pairs. And yet, look how cute he is. This plant, Arabidopsis, has a genome of 135 million base pairs. E. coli, a common bacteria found in our gut, has 4 million base pairs of DNA. These are the ones that if you get the bad ones, make you sick. Chimpanzees have 3.35 billion base pairs. By the way, that's a photo of me and my little brother back in the day. Sorry, I keep jumping to the next one. Mice have 2.5 billion base pairs of DNA. Cats have 3 billion base pairs of DNA. Dogs have 2.4 billion base pairs of DNA. I don't know if you remember, I think I told about this in the first day of school. This was my dog Shotzi that had the broken back, and she lived in that little wheel cart thing for 7 years. Yeah, those carts are amazing. She got around like she never knew the difference. But anyway, that's my dog Shotzi. I still miss that dog. She died like 19 years ago now. Still miss her. Humans. And you could make an argument that this guy in the picture is a human. I guess you could argue that maybe he's not. But anyway, we have, 3.2 billion base pairs of DNA in our genome. That was back when I was healthy. 3.2 billion base pairs. I gotta have some more of my shaved ice. Hang on. So if you laid 3.2 billion base pairs of DNA in our genome, if you laid one copy of the human genome from one cell, if you stretched it and laid it out end to end, it would be about 3.25 feet long. But we have 2 copies of our genome in our cells, except the gametes, right? Therefore, we have about 6.5 feet of DNA in one cell. And an adult human, sorry, back to my shaved ice. Sorry about that. Have about 50 to 100 trillion cells inside our body. So let's use a smaller estimate, right? And let's go with 50 trillion cells. If you take 50 trillion, multiply it times 6.5 feet, that's 325 trillion feet of DNA. And if we divide that by 5280, how many feet in a mile? That's 61.5 billion miles. So if we took the DNA from our entire bodies and laid it end to end, we'd have about 61.5 billion miles of DNA inside of us. And if we use that larger estimate of 100 trillion cells, that would double that. But we'll stick to the 50. 61.5 billion miles of DNA inside little old us. Let's put this in perspective. The moon's about 238,000 miles from the Earth. You take 61.5 billion divided by that, it's 258,000. So there's enough DNA in one human to stretch from here to the moon about 258,000 times. I hope that amazes you, because every time I see this, it still amazes me. 258,000 times from just one person. What about the sun? It's 93 million miles from us. On average, right? You take that number, divide by 93 million, it's 661. So there's enough DNA in one human to stretch from here to the sun 661 times. Again, I hope that amazes you, because that is flat out amazing. So we can go to the moon 258,000 times and to the sun 661 times with the DNA from just one person. Now, consider how amazing this becomes when we look at the weight of our DNA. So we have 3.2 billion base pairs with 3.25 feet of DNA per genome. And it weighs only 3.27 picograms, which is 3.27 times 10 to the minus 12th grams, or that number right there with all those zeros. We have and if you equate from grams to pounds, it's that number right there with all those zeros, 72 pounds. We have 2 copies of our genome per cell, so now we have to double that, right? We end up with all these numbers here, zeros, and then 144 pounds. So that combined weight of the entire genomic DNA from one person, if you take that times 50 trillion cells, if we're dealing with this number here, we talk about it in grams, and take that times 50 trillion, that's 327 grams, or 0.72 pounds of all the DNA from one person. So all that DNA then go from here to the moon 258,000 times, here to the sun 661 times, only weighs about 327 grams, or about 2/3 of a pound, this 0.72 pounds. And again, I hope that flat out amazes you, because it certainly amazes me. So DNA, like I said before, is considered a double helix. Both strands are wrapped around each other like a spiral staircase, or a candy cane is another good way to picture it, where the red colors are spun around in there. So in order to spin that around, you have to imagine that our DNA is like made of rubber or something, so we can wrap it around, or like if you use a rubber band to spin it around each other. And one rotation of the DNA has 10 base pairs in it. So from here to here, it would have 10 base pairs in it. This is only showing 5 of those, but in reality it would be 10 base pairs in between the spinning, the complete rotation of those. But that's not the end, because the DNA doesn't remain linear, because the amount we have Remember, it's 3.25 feet long. That's got to fit inside the nucleus of a cell, right? So it has to overcome this storage dilemma by undergoing a lot of packaging and condensing. And this is kind of an overview of all the packaging and condensing that it goes through, ending up with what we call chromosomes. So we're going to go through this now. Chromosomes are how all the genomic DNA is divided up into smaller sections, typical in higher level organisms like us, but not bacteria. They don't have those. They're not divided up like that. So we're talking mostly about higher level eukaryotes, so like us. Chromosomes come in pairs. So you have 2 copies of each. Like we said, you have 2 copies of each genome. And that's because you get one from your mom and one from your dad, right? Each copy from your parents comes as a complete set of all of your chromosomes. But the chromosome pairs are not identical. Each one has some minor bits that are unique. And that's where the DNA sequence is different. This is all referred to as genetic variation or polymorphisms. And these are all the things that make us all unique and how we look and all that kind of stuff. So just like with genome size, how many chromosomes there are depends on the organism. So I'm going to go through all those same guys. So this lungfish has its 130 billion base pairs divided up into 14 chromosomes. And remember, it comes in 14 pairs, or 28 altogether for both copies. And our cute little pufferfish, he has his 385 million base pairs divided into 21 chromosomes, of which he would have 2 of each, right? 2 copies of each. This Arabidopsis plant only has 5 chromosomes. So he would have 5 pairs, or 10 altogether. E. coli doesn't have any of this. Remember, this is a bacteria. He just has one long piece of DNA. Poor guy. Don't you feel sorry for him? Chimps, as well as all the great apes, have their 3.35 billion base pairs divided into 24 chromosomes. So they would have 2 pairs or 48 for both copies. Mice has all their DNA divided up into 20 chromosomes. So they'd have 20 pairs or 40 altogether. Cats have 19 chromosomes or 38 pairs for all of their 3 billion base pairs. Dogs have 39 pairs or 78 copies, right, for both copies, with their 2.4 times 10 to the 9th base pairs divided up into those 39 chromosomes. Just shows why dogs rule, right? Of course they do. Us humans have our 3.2 times 10 to the 9th base pairs of our genome divided into 23 chromosomes. So we have 23 pairs, or 46 for both copies, right? So we have 23 pairs, or 46 in total. The one pair is called the sex chromosome, determines the sex of the person. Males, us boys, have one X and one Y. You ladies have 2 X chromosomes. Then the other 22 pairs are called autosomal chromosomes. You think I'm supposed to stop here? I think this is where I'm supposed to stop. I think that was a pretty straightforward message, don't you? meaning me, it's not so subtle. So I think we'll stop there. So folks, however far you got today, that's fine. And then I'll check with you when I get back to see how far you got. And if we need to finish this up when we get back, that's what we'll do on the, like I said, I think it's the 19th. So, of course, I had to get one more cough in, right? So with that said, I hope you all have a wonderful day. And I will see you when I come back on the 19th. So let me come over here and shut this down. There we are again. So bye, everybody. Have a wonderful day, and we'll see you back on the 19th. I got to put my glasses on because I can't see where it says stop. There it is. Stop recording. All right. Bye, guys.